Welcome to another video explaining the universe using the particle model. Well, this is the third video covering the senses. I've done two of them before. This is on sound, how hearing happens. There's a lot to talk about, and so I've decided to split this into parts. And this is part one. There will be a part two, hopefully not a part three. But anyway, this is how hearing happens. I've been using the Howard Hughes Medical Institute presentation that was done in 1997 on senses and sensitivity. You can find this series of 4 videos at this one location. And I'm talking now about the third one. The first 2 videos I did the first one was on transduction, getting the message. The one before this was on sight, getting the picture. And yes, this is on how hearing happens. So we get right into the meat of it, where in our ear, inside the inner ear, we have hair cells. This is a cutaway slice of a hair cell showing you the shape and size of it. This is the whole cell right here. And on top of the cell, connected to it, is a hair bundle. And there are neurons leaving this, essentially going down to the brain. This is an artist's drawing of the same thing. This is the whole hair cell. And on top is the hair bundle. All but one of these elements here are called stereocilia. And there's one unique one called kinocilium. And these pieces are joined together and held together and work as a unit. They actually bend back and forth as we get sound coming into our ear. The cell itself actually has a nucleus, which is much more complex than an atom, nucleus of an atom, because it has to do with this whole hair cell. And at the bottom, there are 2 synapses, that is, connections to the brain. This one, there's an arrow pointing down, which suggests that this is when these move, there's a signal sent. Now, this is never discussed in detail. The author presenting this, the speaker in the video from Howard Hughes Medical Institute, talks about a signal being generated as these things move back and forth. How they get from here to here is not clear. However, moving things back and forth like this, where these things, they kind of slide back as it moves, they slide by each other. Those are interactions. Those are mechanical interactions which can release electrons or G1 particles that flow in here. And this is called the afferent, which is the sensatory nerve. They have one coming back. This is the efferent, or the inhibitory synapse, because these are synapses. And there's no information on this. I didn't look it up, but obviously there is some control mechanism. Some inhibitory would imply some sort of throttling, much like a governor on an engine. So that's the hair cell in the ear. This is the speaker in the video, and he had this model of the hair bundle on his desk. And this is when the hair bundle is sitting straight and not being moved by air pressure. He had a model of the ear on the table as well. And then later, shortly after that, he shows how it pushes together and how these move. You have different length of these stereocilia. from short all the way up to tall, and they slide back and forth by each other, and they normally stay straight. They don't bend. The bending is down here, and the sliding is here. And in that whole process of that, my guess, because it's not stated clearly, and I haven't found it on the internet, but a current ends up being flowing through the neuron. It's a hair bundle. So this is the whole ear. This is the outer ear, the pinna. We have an ear canal, which leads to the eardrum. That eardrum goes back and forth, and connected to the eardrum is the hammer. There's a hammer, there's the anvil, and the stirrup. Those are the three tiny bones that you may have heard about when you took science in school. And as the eardrum moves back and forth, it ultimately pushes the stirrup back. The stirrup pushes the cochlea, which is this wound up thing. It looks like a snail. And there are, I think, there's three canals, although they're pointing out only two canals that are parallel, so to speak, inside this. And they cut away one part of these canals to show you not very clear for you, I'm sorry, but the hair-like receptors are inside there. Okay. Well, when I've thought about the hammer anvil, I was always thinking the hammer was hitting the anvil. I mean, the old blacksmith in the old days, working on making horseshoes and whatever, used a hammer to hit the anvil. Now, these are the presumably Latin words malus, like mallet instead of hammer, incus, and stapes. But our common term is hammer, anvil, and stirrup. And you can see how this looks like a stirrup. And interestingly, it's made like this, like it could have more pressure this way and less this way. Whether that's really true or not, I'm making this up just by looking at this and what might happen. But I don't think when you look at how the eardrum being attached to here and the eardrum moves this way and it moves that way, that a concept of a hammer hitting an anvil doesn't work. So I searched around and I found this drawing. Whether it's right or wrong, I found it looks more like a ball and socket. A ball here, socket there. And that would make much more sense. So as the drum moves, the eardrum moves to the left, it causes the whole thing to move to the left, and so it works as a unit. It may have a connection here, so that there's some flexibility on the direction of the force. Hammer, and anvil, and stirrup. Okay, so this is the cochlea. It's wound up. It looks like a snail. It winds up this way, getting small, getting tiny. It winds to the middle. It is 33 millimeters long when it's unwound. And these are the four states that the speaker in the video talks about. Eardrum, hammer, anvil, and stirrup. You can barely see it, but it's a stirrup. But what's important here is it's connected to the top half, or probably one half of the cochlea, and not connected to the other half. And schematically, this is shown in 2 dimensions. And without any change in pressure, without any movement, it just sits there. And this middle line is the basilar membrane. And nominally, it's conceived as being flat. But when you get air pressure, when you get pressure pushing in on the ear, this moves in. You can see that the stirrup is in further. This squishes this outward, which in effect pushes the membrane down. Pressure in, membrane down. Pressure out. Well, it isn't pressure out so much. It's a reduced pressure here allows the internal pressure of your ear to push this out. I contend that this mechanically, these things are all pushing pushing against the internal pressure, and this is the internal pressure pushing against the reduced external pressure. So, but in this case, it moves out, and this thins, causing this to move up. So low pressure, the membrane moves up. High pressure, it moves down. And if you have a continuous signal back and forth, the membrane sits there and vibrates up and down. But now they're going to show you how it does that. I split this into two, showing the basilar membrane. The speaker in the video says that this is basically thinner and narrower. This is basically thicker and wider. And it's not necessarily linear, and I don't remember what all this was about, but that was just generally to show that the shape is not straight. And what this does is it causes the membrane to respond to frequencies difference. Now, these are all audio signals. This is an audio signal at 100 hertz, and this is out in the narrow part of the cochlea. 1,000 hertz is in the middle. 10,000 hertz is towards the thicker part. And he tells us in the video that the ear can hear up to 20,000 hertz. Well, that's the bandwidth of our ear. And so 20,000 has to occur out here. This is if you were getting one pure tone. This is a pure 1,000 hertz. This is 10,000 hertz. This is a composite, presumably of those same three. And this is how the membrane vibrates under a composite signal. One of the interesting things to note is that between the 100 and 1,000, there are 900 hertz change. There's 900 frequencies in here. Well, here there's 9,000 frequencies. So this thing isn't linear at all. There's more room for the low frequency, and there's less room for the high frequency. Being a circuit designer for telecommunications, especially switching, I'm fully aware that the frequency that the telephone switch originally designed for was only 4,000 hertz. So the frequency was here, and we gave more attention to the low frequency to make it more accurate than the low volume that we did for the high frequency. That's because voice only goes no more than 4,000 hertz, whereas sound, music, can go up to 20,000. So let's take a look at do some sort of analysis. This is my analysis. I didn't find this anywhere. The speaker in the video said that there are 16,000 hair cells approximately in the ear. And he claims it's not very many compared to many other cells in the body, including hair cells. There's a lot more than that. This is not that many, and yet we can hear all these frequencies. What I found interesting is that the 16,000 hair cell matches very well with the 2,000 hertz bandwidth, almost like each hair cell was 1 hertz of frequency. So I played the numbers game. I took the 33 millimeter distance of the cochlea, divided it by 20,000, and assumed that there is an average. This is assuming it's linear. It's not linear. The low frequency has maybe twice this, and the high frequency smaller. So this is more of an average of 1.5 micrometer per 1 hertz. It's just an interesting perspective about how many hair cells, what the bandwidth is, and what the physical allocation might be of each frequency in that cochlea. Okay, so we talked about the fact that the membrane moves up and down as the eardrum moves back and forth. Excuse me. And this is one of the slides that the speaker presented. In this case, he's talking about a decrease in pressure, which moves the basilar membrane up. And consequently, the hair bundle moves to the right. And this is trying to show that mechanically, this bottom layer is a basilar membrane. When there's decreased pressure, the internal pressure pushes the drum out against that decreased pressure. The membrane goes up and this whole structure goes up. And the way he presents it here is this very unique structure here where the hair cells, and he's showing 4 of the hair cells, and they're, I'm assuming, straight up in this case, and this is flat relative to the membrane. But when the drum moves out, this moves up, and there's a sliding motion against this tectoral membrane, which causes the hair cells to move to the right. The hair bundle moves to the right. When the eardrum goes out, moves to the left when the eardrum moves in. So this is how these things move back and forth. Now this is an artist drawing, and I'm assuming this are the 3 canals that it has. And this is a group of hair cells. This is that tectoral membrane. And they are generally attached. There's 4 hair cells. At the bottom of the hair cell, it gets to the neurons, to the brain. But it also contains this triangular structure here. It's there. This drawing doesn't show it going very far, but it shows it flat. And so as this moves up, these bend. This is a different picture of the same thing. So what happens when when you get a sound? What happens to it here? Did I mess up someplace here? I lost a slide. There's a slide with a video and that generates this pattern. I apologize for that. If you look at the introduction, when you first open it, there's a description of what there is. I will have a link to a video that has this picture, and you will hear sound as well as how these things change. And of course, this is all graphics, probably mathematically generated, but the speaker in the video, he plays this video. And you can see here's a high frequency at this end and low frequency at that end. And there is a constant pressure on the eardrum. Then the hair bundle moves back and forth to make a sine wave. And it turns out that when the first thing you hear to your ear is a pressure, then that's you don't normally get. You would expect a pressure first. Well, that's the way they drew it. You can see that you get pressure in, causes it to go down and then up. And it causes it to go down first and then up. And so this is a composite tone, and it's all happening at the same time. Individual signals, these signals are being sent to the brain at the same time. Now, this is a picture, a slide I showed on the first video on the senses, had to do with the olfactory senses. And I was explaining how this is the voltage that the presenter was telling would be sent to the brain when an odor molecule was trapped by a receptor in your olfactory senses, and it's like another hair. And I was explaining how it would generate G1 particles on first contact, and at maximum contact you'd get a bunch, and then later a little bit fewer, then maybe none, and then maybe some. It's trying to explain this curve using a G1 particle stream. Same thing must happen in the ear, but the presenter doesn't give us a picture, so I'm making it up. So I start with just a sine wave. I got this off the internet, and it's a sine wave that starts at 0, goes to +5, then -5, then +5. And if this is current flow, then you have electrons flowing. You have a +5 electrons and then a -5 electrons and a +5. Well, wait a minute. If it's, and G1s is the same. That's 0 flowing, 5 flowing, a -5. You can't have a -5 number of electrons flowing towards the brain. It just can't happen. So I redrew it. So on the basis that there is a constant pressure, 14.7 psi, on our ear. It's constant, except it varies a little. So it's constant, so they're probably, with that little varying, it's going to send a stream of G1s, continuous G1s, my opinion, to the brain. Then when the pressure increase, there's an increase. That's what that says, increase, supposed to say increase. The membrane goes down. And then when there's a decrease and the drum goes out, the membrane goes up, and another increase. The scale is different. This lower end is not negative. The lower end is now 0. The average is 5. The peak is 10. So this is 10 G1s. This is 0 G1s. This is 5. Okay, this is just arbitrary, made up, but my guess it's something like that. Okay, well, this is the end of this first part. I'm going to continue on. I didn't want to make this video too long. Part 2 will be next, and I'm going to cover sampling theory, how the signals are combined from the cochlea in the brain, and not, well, at least I'm going to suggest that. And then there's a very interesting thing that the speaker in this particular video talked about, which involves the fact that their ear is not really linear in all cases. There's a non-linear transformation being made, and it has a very unique effect. And I'll be talking about that next time. My name is Bob D. Hilster, and I am your particle model guru. Tune in next time when I'll explain more of the universe using the particle model. Thank you for your attention.